Welcome back to our channel. We hope you will have a wonderful learning experience here with us. You can download and listen to these audiobooks at your preferred time. Our videos are designed as audio podcasts to help you keep studying at times where you may not be able to watch video or as you are completing another task. So, you can better use your time. You can listen to these tutorials at night, doing another task, while taking a walk, lunchtime or at any of your convenience. This is the third part of freshman general biology course taught in all the Ethiopian universities. This chapter deals with the cellular basis of life. It is tailored exactly to the fitting of the Ethiopian Ministry of Education curriculum but is also very useful for any related course. I am very happy you have chosen this channel in your study and politely ask that you subscribe, like and share the channel for more amazing tutorials. Now, without further ado, let's begin our course. Chapter 3, The Cellular Basis of Life. In the late 1600 AYs, an English scientist named Robert Hooke was the first to observe plant cells with a crude microscope. Then, almost a century and a half later, in the 1830 AYs, two German scientists proposed that all living things are composed of cells. Their names were Matthias Schlitten and Theodor Schwann. A German pathologist, Rudolf Virchow, extended this idea by contending that cells arise only from mother cells. 3.1 The cell theory. A cell is the basic structural and functional unit of living organisms. The activity of an organism depends on both the individual and the collective activities of its cells. According to the principle of complementarity of structure and function, the biochemical activities of cells are dictated by their shapes or forms, and by the relative number of their specific subcellular structures. All cells arise from pre-existing cells. Continuity of life from one generation to another has a cellular basis. A typical eukaryotic cell has three major parts. 1. The plasma membrane, the outer boundary of the cell. 2. The cytoplasm, the intracellular fluid packed with organelles, small structures that perform specific cell functions. 3. The nucleus, an organelle that controls cellular activities. Typically the nucleus resides near the cell's center. 3.2 Cell organelles. An organelle is a specialized subunit within a cell that has a specific function. In eukaryotes, an organelle is a membrane-bound structure found within a cell. Just like cells have membranes to hold everything in, these many organs are also bound in a double layer of phospholipids to insulate their little compartments within the larger cells. Prokaryotes are cells that do not have membrane-bound organelles. You can think of organelles as smaller rooms within the factory, with specialized conditions to help these rooms carry out their specific task, like a break room stocked with goodies or a research room with cool gadgets and a special air filter. These organelles are found in the cytoplasm, a viscous liquid found within the cell membrane that houses the organelles and is the location of most of the action happening in a cell. There are two kinds of cell organelles on the basal membrane covering: membranous and non-membranous organelles. Endoplasmic reticulum, rough and smooth; Golgi bodies, mitochondria, chloroplasts; nucleus, lysomes; peroxisomes and vacuoles are membranous, whereas non-membrane-bound cell organelles are ribosomes, 70S and 80S, centrosomes, cilia and flagella, microtubules, basal bodies and microfilaments. 3.3 Structure and function of organelles. The nucleus, this is distinctly oval or spherically shaped largest central structure surrounded by a double-layered membrane. In the nucleus, DNA directs protein synthesis and serves as a genetic blueprint during cell replication. DNA gives codes, or instruction for directing synthesis of specific structure and enzymes proteins within the cell. By monitoring these protein synthesis activity, the nucleus indirectly governs most cellular activities and serves as the cell's master. 3 types of RNA are involved in protein synthesis. At first, NOY's genetic code for a particular protein is transcribed into a messenger RNA, which leaves nucleus through the nuclear pores of the nuclear membrane, and, within the cytoplasm, mRNA delivers the coded message to the ribosomal RNA, which reads message, code and translates it into the appropriate amino acid sequence for the designated protein being synthesized. Finally, transfer RNA transfers the appropriate amino acids within the cytoplasm to their designated site in the protein under production. During cell replication, DNA ensures the continuing of the identical types of cell line within the body. Furthermore, in the gametes, the DNA blueprint serves to pass the genetic characteristics to future generation. Generally, the nucleus may be a sense rounded as in hepatocytes, a sense indented, segmented, as in neutrophils, a sense binucleated, as in parietal cells, cardiac muscle cells, a sense multinucleated as in osteoclasts, skeletal muscle cells, a sense very large, many DNA, as in megakaryocytes, a sense absent, as in mature erythrocytes, blood platelets. The nucleus is surrounded by a nuclear envelope and contains chromadin and one or more nucleoli. The nuclear envelope surrounds nuclear material, consists of outer and inner membrane, perforated at intervals by nuclear pores. Through this pores most ions and water-soluble molecules to transfer N nucleus and cytoplasm. Chromadin. The term chromadin means colored material and refers to the fact that this material is easily stained for viewing with microscope, and it is composed mainly of coils DNA bound to basic protein called histones. The DNA is so long that it has to be packed in an organization manner or it would get entangled like an unraveled ball of string. Nucleoli. The nuclei of most cells contain one or more lightly stained structures called nucleoli that actively engage in synthesizing of ribosomes. The nucleolus, unlike most of the organelles, does not have a limiting membrane. Instead, it is simply a structure that contains large amounts of RNA and protein of the type found in ribosomes. The nucleolus becomes considerably enlarged when a cell is actively synthesizing proteins. The genes of 5 separate chromosome pairs synthesize the ribosomal RNA and then store it in the nucleolus, the cytoplasm. The cytosol is the material of cell interior not occupied by the nucleus, containing a number of distinct, highly organized membrane-enclosed structures. The organelles dispersed within a complex jelly-like marrow called the cytosol. All cells contain 6 main types of organelles: the endoplasmic reticulum, Golgi Complex, Lysomes, peroxisomes, mitochondria, and vacuoles. They are similar in all cells, but with some variations depending on the cell specialization. Each organelle is a separate compartment, containing different chemically setting for fulfilling a partial or cellular function. These organelles occupy about half of the total cell volume. The remaining part of the cytoplasm is cytosol. Endoplasmic reticulum, The endoplasmic reticulum is a fluid-filled membrane system extensively present throughout the cytosol. The is one continuous organelle with many communicating channels. The 2 different types are smooth endoplasmic reticulum and the rough The smooth is a meshwork of interconnected tubules, whereas the rough projects outwards from the reticulum as stacks of flattened sacs. Though different in structure and function, they are continuous with each other. The rough endoplasmic reticulum, the outer surface of the rough contains dark particles called ribosomes, which are ribosomal RNA protein complexes that produce protein under the direction of nuclear DNA. Messenger RNA carries the genetic message from the nucleus to the ribosomes workshop where proteins are synthesized. Some ribosomes are free dispersed throughout the cytosol. The rough in association with ribosomes produces and releases a variety of proteins into the fluid-filled space enclosed by the membrane. Some proteins for export as secretory products, hormones or enzymes. Other proteins are transported to sites within the cell for use in the construction of new plasma membrane or new organelle membrane. Cellular membrane contains predominantly fats and proteins. Membrane also contains enzymes required for the synthesis of almost all the lipids needed for the production of new membranes. These lipids enter the lumen along with the proteins. This structure is well developed in cells producing digestive enzymes or in rapidly growing cells. Each ribosome is involved in producing only one type of protein. The free ribosomes synthesize enzyme protein that are used intracellularly within the cytosol. Smooth endoplasmic reticulum. Since it does not have ribosomes, it looks asmooth and does not produce proteins. It serves a variety of other functions that differ in cell types. In most cells, the smooth ER is sparse and serves in packaging and discharging site for protein molecules that are to be transported from the ER. All new proteins and fats pass from ER gathered in the smooth ER. Portions of the smooth ER then bud off, pinch off, giving rise to A's transport vesicles OY. They contain the new molecule wrapped in a membrane derived from the smooth ER membrane. Transport vesicles move to the Golgi complex for further processing of their cargo. Some specialized cells have an extensive smooth ER, which has additional functions as follows. The smooth ER is well developed in cells specialized in lipid metabolism, cells that synthesize steroid hormones. The membrane wall of the smooth ER contains enzymes for synthesis of lipids. This is an additional site for synthesis in addition for ER to keep pace with demands for hormone secretion. In liver cells, the smooth ER contains enzymes involved in detoxifying harmful endogenous substances produced within the body by metabolism or exogenous substances entering the body from outside as drugs or other foreign compounds. The detoxifying enzymes alter toxic substances so that they could be easily eliminated in the urine, but unfortunately, in some instances the same enzyme transforms otherwise harmless substance into carcinogens that play a role in cancer development. The smooth ER has a special role in skeletal muscle cells. They have an elaborate network of smooth ER, which stores ionic calcium and plays a crucial role in the process of muscle contraction. The Golgi Complex. The Golgi Complex is elaborately associated with the ER and contains sets of flattened, curved, membrane-enclosed sacs, or cisternae, stacked in layers. Number of stacks vary in cells. Cells specialized for protein secretion have hundreds of stacks, whereas some have only one. The majority of newly formed molecules budding off from the smooth ER enter a Golgi complex stacks. It performs the following important functions. 1. Processing the raw material into finished products. In the Golgi complex, the raw protein from the ER are modified into their final state mainly by adjustment made in the sugar attached to the protein. This is a very elaborate, precisely programmed activity, specific for each final product. 2. Sorting and directing finished product to their final destination. According to their function and destination, different types of products are segregated by the Golgi complex, that is, molecules that are destined for secretion to the exterior, molecules that will eventually become part of the plasma membrane, and the molecules that will become incorporated into other organelles. 3. The smooth ER of the liver and kidney cells are responsible for the detoxification and inactivation of drugs. Enzymes within the smooth ER can inactivate or destroy a variety of chemicals including alcohol, pesticides, and carcinogens. 4. In skeletal muscle cells, a modified form of smooth ER stores Ca2+ to be released for muscle contraction. Lysomes. Lysomes serve as the intracellular digestive system. Lysomes are membrane-enclosed sacs containing powerful hydrolytic enzymes capable of digesting and removing unwanted cellular debris and foreign materials such as bacteria that have been internalized within the cell. Lysomes vary in size and shape, and about 300 I circumflex 1/4 m in a cell. Surrounding membrane confines these enzymes, preventing from destroying the cell that houses them. Extrinsic material to be attacked by lysosomal enzymes is brought into the interior of the cell through the process of endocytosis. If the fluid is internalized by endocytosis, the process is called pinocytosis. Endocytosis is also accomplished by phagocytosis. In pinocytosis, ECF and a large molecule such as protein is engulfed. A specific molecule may bind to surface receptor, triggering pinocytosis receptor-mediated endocytosis. Dynamin, a molecule forms rings wrapping around, severing the vesicle from the surface membrane in pinocytosis. In phagocytosis, large multimolecular particles are internalized by endocytosis. This is achieved by only a few specialized cells, white blood cells, that play an important role in the body's defense mechanism. When a leukocyte encounters large multimolecular particle, such as bacteria or tissue debris, it extends projection, pseudopodia, that completely surround or engulf the particle, forming an internalized vesicle that traps the large multimolecular particle within it. A lysome fuses with the membrane of the internalized vesicle and releases its contents of hydrolytic enzymes into the vesicle. These enzymes safely attack the microbes or other trapped material within the enclosed confines of the vesicle without damaging the remainder of the cell. Lysomes can take up whole organelles such as mitochondria and break down into their component molecules. Those molecules that can be released are reabsorbed into the cytosol, and the rest are dumped out of the cell. The process by which worn-out organelles are digested is called autophagy. A human liver cell recycles about half its content every week. In the inherited condition known as lysosomal storage disease, Tay-Sachs disease, lysosomes are not effective because they lack specific enzymes. As a result, harmful waste products accumulate, disrupting the normal function of cells, often with fatal results. Peroxisome. Peroxisome is membrane-enclosed sacs containing oxidative enzymes and catalase that detoxify various wastes. Oxidative enzymes need oxygen to remove hydrogen from specific substance molecules. Such reactions are important in detoxifying various waste products within the cell or foreign compounds that have entered in, such as ethanol consumed in alcoholic drinks. In liver and kidneys, the major product generated is hydrogen peroxide. Hydrogen peroxide itself is a powerful oxidant. It also contains catalase, an antioxidant enzyme decomposing hydrogen peroxide into harmless water and oxygen. This reaction is an important safety reaction that destroys deadly hydrogen peroxide at the site of production, thereby preventing possible devastating escape into the cytosol. It is shorter and smoother than lysosome and several hundreds may present in one cell. Peroxisomal disorders disrupt the normal processing of lipids and can severely disrupt the normal function of the nervous system by altering the structure of the nerve cell membrane. Mitochondria. Mitochondria are the powerhouses of a cell. They extract energy from nutrients and food and transform it into usable form to energize cell activity. Their number varies depending on the energy needs of each particular cell types. A single cell may have few hundreds or thousands. Mitochondria are rod or oval shaped about the size of a bacterium. Each is enclosed by a double membrane, a smooth outer that surrounds the mitochondria, and an inner membrane that forms a series of unfolding or shelves called cristae, which project into an inner cavity filled with a jelly-like matrix. See Figure 3.5. These cristae contain proteins that convert much of the energy and food into a usable form. The electron transport protein, the unfolding increase the surface area available for keeping these important proteins. The matrix contains a mixture of hundreds of different dissolved enzymes, citric acid cycle enzymes, that are important in preparing nutrient molecules for the final extraction of usable energy by the cristae proteins. Carbon-hydrogen bonds in ingested food are the source of energy stored in the chemical forms. Body cell can extract energy from food nutrients and convert it into energy form that they can use. The high-energy phosphate bonds of ATP contain adenosine with 3 phosphate groups. When high-energy phosphate bond is split, a substantial amount of energy is released. ATP is the universal energy carrier, the common energy currency of the body. Cells can cash in ATP to pay the energy price for running the cellular machine. To get immediate usable energy, cells can split terminal phosphate bond of ATP, which yields ADP with phosphate group attached, plus an organic phosphate, Pi, plus energy. Mitochondria are unusual organelles in 2 ways. 1. In the matrix they have their own unique DNA called mitochondrial DNA. 2. Mitochondria have the ability to replicate themselves even when the cell to which they belong is not undergoing cell division. Chloroplasts. Chloroplasts are useful organelles among plastids as they highly participate in the process of photosynthesis, which is a process by which plants synthesize their own food. They are located in outer surface of the cell to receive enough light. Chloroplasts are green-colored due to the chlorophyll pigments found in its internal parts. Some of important characteristics of plant is its ability to carry out photosynthesis as the way they use in making their own food and pass through converting light energy and chemical energy. Vesicles are membrane-bound sacs that are used to store or transport substances around the cell. Lysomes are actually vesicles. Vacuoles are essentially larger vesicles, and they are formed by the joining together of many vesicles. They are membrane-bound organelles that have no specific shape and contain water with a number of different compounds within it. Their function varies greatly depending on the type of cell they are part of. In plant cells they are important in maintaining turgor pressure. Cytoskeleton. The cytoskeleton is a complex protein network that acts as the bone and muscle of the cell. This necessary intracellular scaffolding supports and organizes cellular components' arrangements and to control their movements. This provides distinct shape, size to the cell. This network has at least 4 distinct elements: microtubules, microfilaments, intermediate filaments and microtubular lattice. The different parts of the cytoskeleton are structures linked and functionally coordinated to provide integration of the cell. The microtubule is the largest of the group. Slender, long, hollow tubes composed of a globular protein molecule, 6 nanometers diameter, tubulin. They provide a symmetrical shape to the cell, such as a neuron with cell body and long axon. They coordinate numerous complex cell movements and transport of secretory vesicles from region to region of the cell, movements of cilia and flagella, distribution of chromosomes during cell division. Microfilaments are important to cellular contractile system and as mechanical stiffeners. The microfilaments are the smallest of the cytoskeleton, composed of protein molecule actin having a globular shape similar to tubulin. Generally, cytoskeletons determine/provide the shape of a cell, structural support, organizing its contents, substances, movement through cell, cilia, flagella and intracytoplasmic vesicles, and contribute to movements of the cell as a whole. Plasma, cell membrane. The plasma membrane is extremely thin layer of lipids and proteins forming outermost boundary of living cell and enclosing the intracellular fluid, ICF. It serves as a mechanical barrier that traps needed molecules within the cell. Plasma membrane plays an active role in determining the composition of cell by selective permeability of substances to pass between the cell and its CCF environment. There are some differences in the composition of plasma membrane between cell types, which permits the cell to interact in different ways with essentially the same extracellular fluid, ECF, environment. The plasma membrane is a fluid lipid bilayer embedded with proteins. It appears as trilaminar layer structure having 2 dark layers separated by a light middle layer as a result of specific arrangement of the constituent molecules. All plasma membrane are made up of lipids and proteins plus small amount of carbohydrates. Phospholipids are most abundant with a lesser amount of cholesterol. Phospholipids have a polar charged head having a negatively charged phosphate group and 2 nonpolar, electrically neutral, fatty acid tails. The polar end is hydrophilic, water-loving, because it can interact with water molecule, which is also polar. The nonpolar end is hydrophobic, water-fearing, and will not mix with water. Such 2-sided molecules self-assemble into a lipid bilayer, a double layer of lipid molecules. When in contact with water, the hydrophobic tails bury themselves in the center away from the water, while the hydrophilic heads line up on both sides in contact with water. The water surface of the layer is exposed to ECF, whereas the inner layer is in contact with the intracellular fluid, ICF. The lipid is fluid in nature, with consistency like liquid cooking oil. Cholesterol provides to the fluidity as well as the stability. Cholesterol lies in between the phosphate molecules, preventing the fatty acid chain from packing together and crystallizing that could decrease fluidity of the membrane. Cholesterol also exerts a regulatory role on some of the membrane proteins. For fluidity of the membrane, it gives flexibility to the cell to change its shape. Transport process are also dependent on the fluidity of the lipid bilayer. The membrane proteins are either attached to or inserted within the lipid bilayer, some extending through the entire membrane thickness. They have polar region at both ends joined by a nonpolar central portion. Other proteins are on either the outside or inner surface, anchored by interactions with proteins that spans the membrane or by attachment to the lipid bilayer. On account of membrane fluidity, many proteins float freely, although the mobility of protein that have special function in a particular area of the membrane is restricted. This gives ever-changing mosaic pattern of the protein embedded in the lipid layer. Only the outer surface of the plasma membrane contains a small amount of carbohydrate. Short-chain carbohydrates are bound primarily to membrane proteins and to a lesser extent to lipids, forming glycoproteins and glycolipids. The plasma membrane is actually asymmetrical. The two surfaces are not the same. Carbohydrate is only on the outer surface. Different amount of different proteins are on the outer and inner surfaces, and even the lipid structures of the outer and inner half is not the same. The plasma membrane is highly complex, dynamic, regional differentiated structure. The lipid layer forms the primary barrier to diffusion, whereas proteins perform most of the specific membrane functions. Lipid bilayer forms the basic structure of the membrane, is a barrier to passage of water-soluble substances between the ICF and ECF, and is responsible for the fluidity of the membrane. Membrane proteins are a variety of different proteins within the plasma membrane have the following special functions. 1. Some form water-filled passageways or channels. Across the lipid bilayer, such channels allow ions to pass through without coming in direct contact with lipid interior. The channels are highly selective. They can selectively attract or repel particular ions. This selectively attracts or repels particular ions. This selectivity is to specific charged amino acids. Group, number and kind of channels vary in cells. Channels open and close in response to a controlling mechanism. 2. Other proteins serve as carrier molecule that transport specific molecule that cannot cross on their own. They differ in cells. For example, thyroid epithelial cell possesses carriers for iodine. 3. Many proteins on the outer surface serve as ACE receptor sites that recognize and bind with specific molecules in the cell environment. This binding triggers a series of membrane and intracellular events that alter the activity of the target cell. In this way, hormones influence specific cell, even though every cell is exposed to the same chemical messenger via its widespread distribution by the blood. 4. Another group of proteins act as membrane-bound enzymes that control specific chemical reactions on either side of the plasma membrane. For example, outer layer of the plasma membrane of skeletal muscle contains enzyme macrosterase that destroys the chemical messenger that triggers contraction. 5. Some proteins are arranged as filaments meshwork on the inner side and are secured to certain internal protein elements of the cytoskeleton. They maintain cell shape. 6. Other proteins function as cell adhesion molecules, CAMs. These molecules protrude from the membrane surface that grip each other and grip the connective tissue fibers that interlace between cells. 7. Some proteins, especially in conjunction with carbohydrate, are important in the cell-wise ability to recognize A-self-Y and in cell-to-cell interactions. Membrane carbohydrate, short-chain carbohydrates on the outer membrane surface, serves as self-identity marker, enabling cells to identify and interact with each other in the following ways. A-sense-diaresis, recognition of self and cell-to-cell interactions. Cells recognize each other and form tissues. Complex carbohydrates act as a trademark of a particular cell type, for recognition. A-sense carbohydrate containing surface markers are important in growth. Cells do not overgrow their own territory. Abnormal surface markers present in tumor cells, and abnormality may underline uncontrolled growth. A-sense some CAMs have carbohydrate on the outermost tip where they participate in cell adhesion activity. Functions of biological membranes. The phospholipid bilayers provides the basic structure of the membrane, and they also restrict entry and exit of polar molecules and ion. The other molecules in the membrane have a variety of function. A-sense channel protein and carrier protein. These proteins are involved in the selective transport of polar molecule and ion across the membrane. A-sense enzymes. Membrane proteins sometimes act as enzymes. For example, the microvilli epithelial cells lining some part of the gut contain digestive enzymes in their cell surface membrane. A-sense receptor molecules. Proteins have very specific shapes, and this makes them ideal as receptor molecules for chemical signaling between cells. For example, hormones are chemical messengers, which circulate in the blood but only bind to specific target cells, which have the correct receptor sites. Neurotransmitters, the chemicals that enable nerve impulse from one nerve cell to the next, also fit into specific receptor proteins in nerve cells. A-sense antigens. These act as cell identity markers or name tag. They are glycoproteins, that is, proteins with branching carbohydrate side chains like antennae. There is an enormous number of possible shapes to these side chains. so each type of cell can have its own specific markers. This enables cells to recognize other cells, and to behave in an organized way. For example, during development of tissues and organs in multicellular organisms, it also means that foreign antigens can be recognized and attacked by the immune system. Glycolipids also have branching carbohydrate side chain and are involved in cell-cell recognition. They may act as receptor sites for chemical signals. With glycoproteins they also involved in sticking the correct cell together in tissues. Sperm recognition of ova sense energy transfer in photosynthesis and respiration. Proteins take part in the energy transfer systems that exist in the membranes of chloroplast and mitochondria respectively. Sense cholesterol acts like a plug, reducing even further the escape or entry of polar molecules through the membrane. 3.4 Cellular diversity. Cells are found in different organisms, and are very diverse in their size, shape and their internal structure, and this also applies to cells found in the same organism. This diversity is influenced by their roles and function within organism-wise body. 3.4.1 Cell shape. Cells have different shapes due to appropriate function. It is possible to find other cells which are flat. Most of these cells are body cells and their function is protecting and covering body surface. Nerve cells have long extensions. Skin cells have a shape which is flat. Egg cells have shape which is like sphere, and some bacteria are rod in shape. Some plant cells are rectangular. 3.4.2 Cell size. Some cell can be seen without using magnification instruments as they are enough to be seen by the naked eye. Example, egg of birds, reptiles and a neuron cell of giraffe, which is 2 meters in length. 3.5 Transport across the cell membranes. The plasma membrane is selectively permeable. Lipid-soluble substances and small ions can passively diffuse through the plasma membrane down their electrochemical gradients. Highly lipid-soluble particles are able to dissolve in the lipid bilayer and pass through the membrane. Uncharged, nonpolar molecules oxygen, carbon dioxide and fatty acids are highly lipid-soluble and readily permeate the membrane. Charged particles sodium, potassium ions and polar molecules such as glucose and proteins have low lipid solubility, but are very soluble in water. For water-soluble ions of less than 0.8 nanometers diameter, protein channels serve as an alternate route for passage. Ions for which specific channels are available can permeate the plasma membrane. Particles with low lipid permeability and too large for channels cannot permeate the membrane on their own. The phospholipid bilayer is a good barrier around cells, especially to water-soluble molecules. However, for the cell to survive, some materials need to be able to enter and leave the cell. There are 4 basic mechanisms. 1. Diffusion and facilitated diffusion. 2. Osmosis. 3. Active transport. 4. Bulk transport. 2 forces are involved in facilitating movement across the plasma membrane. 1. Forces that do not require the cell to. A passive force. 2. Forces requiring energy, as ATP, to be expended to transport across the membrane. Active force diffusion. A diffusion is the net movement of molecules, or ions, from a region of their high concentration to a region of their lower concentration. The molecules move down a concentration gradient. Molecules have kinetic energy, which makes them move about randomly. All molecules in liquid and gases are in continuous random motion in any direction as they have more room to move before colliding with another. As a result of this haphazard movement, the molecules frequently collide, bouncing off each other in different directions. The greater the concentration, the greater the likelihood of collision. Such a difference in concentration in molecules between 2 adjacent areas is chemical/concentration gradient. The net movement of the molecule by diffusion will be from the higher area of concentration to the area of lower concentration. Certain additional factors that influence the rate of net diffusion across a membrane are: 1. Permeability of the membrane. 2. Surface area of the membrane. 3. Molecular weight of the substance. Lighter one diffuses rapidly. 4. Distance through which diffusion must take place. Note that, increasing all the factors increases rate of net diffusion, except distance, thickness, that if increased, decreases the rate of diffusion, and molecular weight if increased, decreases rate of diffusion. As a result of diffusion, molecules reach an equilibrium where they are evenly spread out. This is when there is no net movement of molecules from either side. Movement along electrical gradient. Movement of charged particles is also affected by their electrical gradient. If a relative difference in charges exists between two adjacent areas, the cations tend to move towards more negatively charged area, whereas the anions tend to move toward the more positively charged areas. The simultaneous existence of an electrical and concentration, chemical, gradient for a particular ion is referred to as an electrochemical gradient. Carrier-mediated transport. All carrier proteins span the thickness of the plasma membrane and are able to undergo reversible changes in shape so that specific binding site can alternately be exposed at either side of the membrane. As the molecule to be transported attaches to a binding site on the carrier on one side of the membrane, it triggers a change in the carrier shape that causes the same side to be exposed to the other side of the membrane. There having movement in this way, the bound molecule detaches from the carrier. This transport displays three characteristics. 1. Specificity. Each cell possesses protein specified to transport a specific substance or few closely related chemical compounds. Amino acid cannot bind to glucose carrier, but similar amino acids may use the same carrier. Type of carriers vary in cells. A number of inherited disorders involve defects in transport system for a particular substance. 2. Saturation. In a given time only a limited amount of a substance can be transported via carrier. A limited number of carrier sites are available within a particular plasma membrane for a specific molecule. This limit is known as transport maximum, trademark. The substance's rate of transport across the membrane are directly related to its concentration. When the trademark is reached, the carrier is saturated, and the rate of transport is maximum. Further increase in the substance concentration is not accompanied by corresponding increase in the rate of transport. Saturation of carrier is a critical rate-limiting factor to the transport of selected substances across the plasma membrane and kidney and the intestine. There is a mechanism to increase the number of carriers in the plasma membrane. 3. Competition. Several closely related compounds may compete for ride across the plasma membrane on the same carrier. Facilitated diffusion. Facilitated diffusion uses a carrier protein to facilitate the transfer of a particular substance across the membrane downhill from higher to lower concentration. This process is passive and does not require energy because movement occurs naturally down a concentration gradient. Active transport, on the other hand, requires the carrier to expend energy to transfer its passenger uphill against a concentration gradient from an area of lower concentration to an area of higher concentration. Active transport requires protein carrier to transfer a specific substance across the membrane, transporting against concentration gradient. Carrier phosphorylation increases the affinity for its passenger. The carrier has ATPase activity, splitting high-energy phosphate from an ATP to yield ADP plus a free pi. This phosphate group gets bound to the carrier. Phosphorylation and binding of particle on the low concentration side induces a conformational change in the carrier protein so that passenger is now exposed to the high concentration side of the membrane. This change in carrier shape is accompanied by dephosphorylation. Removal of phosphate reduces the affinity of the binding site for the passenger, so the passenger is released on the high concentration side. The carrier then returns to the original conformation. This active transport mechanisms are often called ACE pumps, analogous to lift water by pump that need energy to lift water against the downward pull of gravity. Hydrogen pump, ACE pump, A pump. Osmosis. Osmosis is the net diffusion of water down its own concentration gradient. Water can readily permeate the plasma membrane. The driving force for diffusion of water is its concentration gradient from area of higher water concentration, low solute, to the area of lower water, high solute, concentration. This net diffusion of water is known as osmosis. Special mechanisms are used to transport selected molecules unable to cross the plasma membrane on their own. Active transport. Active transport is energy-consuming transport of molecules or ions across a membrane against its natural tendency to diffuse in the opposite direction. The movement of molecules in active transport is in one direction only. Unlike diffusion that is reversible, the energy is supplied by the breakdown of ATP, which energy carrier is made in respiration. The major ions within the cells and their surrounding are sodium, Na+, potassium, K+, and chloride, Cl. In recent years it has been shown that the cell surface membrane of most cells have sodium pump is coupled with a potassium pump that actively moves potassium ion from outside to inside the cell. The combined pump is called the sodium pump, Na plus K pump. The pump is a carrier protein that spans across the membrane from one side to the other. The transfer of sodium and potassium across the membrane is brought about by the changes in the shape of the protein. Note that for every 2 K+ ions taken into the cell, 3 Na+ ions are removed. Thus a potential difference is built up across the membrane, with the inner side of the cell being negative. This tends to restrict the entry of negatively charged ions, anions, such as chloride, and favoring diffusion of cations into the cell. This explains why chloride concentration inside red cell is less than the outside, despite the fact that chloride ions can diffuse in and out by facilitated diffusion. Similarly, positively charged ions, cations, tend to be attracted into cell. Thus both concentration and charge are important in deciding the direction in which ions cross the membrane. The pump is essential in controlling the osmotic balance in animal cells, osmoregulation. If the pump is inhibited, the cell swells and bursts because of the building up of Na+, which results in excess water entering into the cell by osmosis. The pump is also important in maintaining electrical activity in nerve and muscle cells and in driving active transport of some other substances such as sugar and amino acids. In addition, high concentration of potassium are needed inside cells for protein synthesis, glycolysis, photosynthesis, and other vital processes. Sodium-potassium pump plays 3 important roles. 1. It establishes sodium and potassium concentration gradients across the plasma membrane of all cells. These gradients are important in the nerve and muscle to generate electrical signals. 2. It helps regulate cell volume by controlling the concentration of solutes inside the cell and thus minimizing osmotic effects that would induce swelling or shrinking of the cell. 3. The energy used to run the pump also indirectly serves as the energy source for the co-transport of glucose and amino acids across the membrane, intestine and kidney cell. Exocytosis and endocytosis vesicular transport. The special cell membrane transport system selectively transports ions and small polar molecules. But large polar molecules and even multimolecular material may leave or enter the cell, such as hormone secretion or ingestion of invading microbe by leukocytes. These materials cannot cross the plasma membrane but are to be transferred between the ICF and ECF not by usual crossing but by wrapped-in membrane. This process of transport into or out of the cell in a membrane-enclosed vesicle is vesicular transport. Transport into the cell is termed endocytosis, whereas transport out of the cell is called exocytosis. In endocytosis, the transported material is wrapped in a piece of the plasma membrane, thus gaining entrance to the interior of the cell. Endocytosis of fluid is called pinocytosis, cell drinking, whereas endocytosis of large multimolecular particle is known as phagocytosis, cell eating. Endocytosis and exocytosis are active processes involving the bulk transport of materials through membranes, either into cells, endocytosis, or out of cells, exocytosis. Endocytosis occurs by an enfolding or extension of cell surface membrane to form a vesicle or vacuole. It is of two types. Phagocytosis, cell eating, material taken up is in solid form. Cells specializing in the processes are called phagocytes and are said to be phagocytic. The sac formed during the uptake is called a phagocytic vacuole. Pinocytosis, cell drink, material taken up is in liquid form. Vesicles formed are extremely small, in which case the process is known as micropinocytosis and the vesicles as micropinocytic vesicles. Pinocytosis is used by the human egg cell to take up nutrients from the surrounding follicle cells. Exocytosis is the reverse process of endocytosis. Waste materials such as solid and undigested remains from phagocytic vacuoles may be removed from cells, or useful materials may be secreted. Secretion of enzymes from the pancreas is achieved in this way. Plant cells use exocytosis to export the materials needed to form cell wall. I hope this was a fruitful study. You should now be able to state cellular bases of life, cell theory, identify organelles, their structure, function, and explain the importance of homogenization in cell study. Thank you for reading with us. Join us again in the next chapters.